state of genome scale metabolic networks in continuous cell culture.
Right. Thank you very much. So I come from Habana. It's okay. It works. It's terrible. All right. That's the title of the talk, and it has actually two parts. The first part, which is the work I started to do when we enter into Internet, and the second part that is what we are starting to do right now, so at the end of the project. So in the first part, I am going to speak about a sort of industrial process, which was important for us in this biotech industry. And the second part is something more fundamental, if you want, about statistical physics and metabolic networks. But you will see that at least methodologically, they are both connected. And then I will talk about the conclusion. So let's start with the first part of the talk. This is my problem. So I am a theoretical physicist, but I have some good people and some friends working in this biotech industry in Cuba that, as you know, is more or less important. And essentially, they have this kind of continuous process in which, they cultivate some cells here, and the cells grow, produce some proteins, and then they remove the cells. And at the same time, they put nutrients to this vessel here with the same dilution rate. Let's say they introduce some nutrients here, cells grow, and at the same time they remove nutrients and cell. And the idea is not to have a batch process in which you give a lot of nutrients to the cells and the cells just grow a lot and produce whatever you want, and then you remove everything and you get the protein. The idea, the dream, if you want, of the people doing this kind of industrial process is to have a continuous system of producing proteins. That would be the task. And therefore, they wanted to optimize this problem. And they, of course, find a lot of problems with this system. One of the problems is that at some moment, cells essentially die. So you have, everything is working perfectly well, and for reasons that are not very well understood, cells die. When they told me about the project, I thought, all right, but we're in Cuba, so surely, something wrong happened at some moment, so the guy that have to work there doesn't go, the air conditioner is not working. So they convinced me that this is not the case, so that they actually take care of it, and something is happening. And then talking with other colleagues around, I realized that this is a common problem when you have a continuous culture. That's something that happens at some point. And then we say, all right, let's try to understand first what is the actual states of the system. So how you can describe properly something like that. So our main variables will be here, the concentration of cells, which is X, and SI, which is the concentration of some metabolites in the vessel. CI is the concentration of some nutrients in the vessel, right? So I is an index 1, 2, 3, 4, 5. You have many nutrients, and those are our variables. And what we like to do is to connect what is happening in the vessel with what is happening inside a cell, right? So we are going to model the metabolism of the cell and to connect this with what is happening on the vessel, right? And these are the equations, very simple. The first one here is a dynamics for what is happening in the vessel, for the number of cells. The number of cells depend on mu, which is the growth rate of the cells, d, which is the dilution rate, so how fast you remove cells, that's why M minus d. And this sigma here is something that we introduced just to take into account the fact that some metabolites may be toxic for the cell. That's the cell parameter there, right? So what is interesting is that, and then we have the second equation that tells you how the concentration of some metabolites change on time, right? There is a first term that essentially tells you that U is the flux of the metabolites, so how fast they consume or produce the metabolites, right? And then you have a minus here because if it's producing, so it goes up, and if it is consuming, it goes down. Then you have again this term here that is essentially connected with the dilution. You have here Ci, that means if it is coming from the outside, you have that the concentration of the metabolites is increasing. So if Ci is some nutrient, so the concentration increase, and minus S because you remove it with the dilution rate D, right? Those are the two equations. First, you have an equation for the number of cells, and then you have an equation for the concentration of the metabolites, right? And of course, they are connected in two ways. First, because this one here depends on the number of cells. If you have more cells, you consume more, for example, and then you have less metabolite at a certain time. Or if they produce the metabolite, the more cells you have, the more metabolites you get. And this is one connection. And the other connection comes from the fact that this mu here depends on what is happening inside the cell. So U here is a vector that describes the internal fluxes of the reactions, and R here, U is a vector that describes the exchange fluxes of the metabolite, of the metabolism, and R is again a vector that describes the internal fluxes within the cell. So essentially what we're going to do is to say, all right, if I have a cell that is growing with a certain mu, that will depend on what is on, then this comes here, I can solve this, I have X, and X come here, and I have to solve this. And of course, you should do everything at the same time. It's self-consistent in some way. And this sigma here is, as I told you, is a measure of the death rate of the system that will be connected with the concentration of some metabolites here. Therefore, this S here can be understood also as a vector. Right? That's more or less a picture. What we have to decide now is what is this mu here. How you calculate, given the cell, what's the growth rate of the cell. That's what we want to do there. And the other thing I want to point out is that, of course, is, for example, if we assume, as we will do, that we are in stationary state, so U is bounded by this reaction here. So if you want to know which are the bounds for the reaction rate, there are some information that the biochemistry gives to you. So, I mean, this reaction cannot be larger than this stuff here because we understand the dynamics or the kinematics of this reaction. But in this particular case, you have another bound that is given by the fact that if S is 0, you cannot consume U at the velocity that is larger than dc divided by x. That's intuitively what is coming out, and that will be relevant now, right? But if everything is clear now, I can proceed and I will explain you just essentially how to solve the problem, right? So what's happening inside the cell, which is the complex stuff. Inside the cell, you have a lot of reaction. We are going to assume that we are in stationary state, so that what's happening in the cell happens faster than what is happening in the vessel. And this is, and this is essentially describing all the reaction inside the cell. So this S here is a stoichiometric matrix. R are internal fluxes. U are exchange fluxes. EI is essentially a maintenance fluxes that you ask for some metabolites. And this term here introduces the growth rate of the system, right? So you introduce the growth rate as a different reaction. And you assume, this is very standard in what is called flux balance analysis theory for metabolism. And then you assume that the fluxes are bounded in some way, and in particular the exchange fluxes are bounded by VI, which is what biochemistry tells you, and by the fact that you cannot violate the constraint of the dynamic reaction I just explained you. So in this way we have a new way to connect what is happening outside the vessel with what is happening inside the cell. So the way in which the cell is understanding what is happening outside is through the fact that some exchange fluxes are bounded by this term here, in which you introduce the X. Right? So we can go a bit back. So here, there is no way, so you don't have any connection between mu and what is happening in the vessel. This is not described here. So the connection comes when you realize that the fluxes, the exchange fluxes have to know somehow how much cells you have in the vessel, and that's the connection, right? And then we are going to make a new assumption, that is an important one, that is that the cells that are in the vessel are maximizing biomass. If this is the case, this set of equations, although very hard, very ugly if you want, are easy to solve because it's just a linear programming technique, right? So the intuition is, we can solve this because we assume, let's put everything here. So on the right, we have what is happening in the vessel in a stationary state. It means that we should solve for mu and for u, and this is something that can be done easily algorithmically. So you have a linear programming problem that given an x, solve you all of this stuff, right? And then you can go back and to solve this dynamical equation, and then to go back and forth until the system is solved. And first we started to study a simple model. Forget about very complex metabolism. Let's try to take a sort of toy model for metabolism, which I think, if it's not the very well established, it's the one we like, essentially, and we have used it during these 2 or 3 papers we have made. And the idea is that you have these simple cells that consume something and produce energy in 2 ways. So this is glycolysis if you want, this is the Krebs cycle, and at the same time they produce waste. And this waste goes as a toxic byproduct. So this is our reaction rate. If you want to go back a bit, this is what you put here, right? You describe this reaction and you put it here, right? And then you solve everything, and you assume that this waste here, that we will call lactate, just because to connect with something realistic, is toxic to the system, right? And just to make it short, that's the kind of picture you get. So you have this, I would say, phase diagram in which you have X and you have the dilution, which are the 2 parameters. And what is interesting is that this is the good part of the phase diagram, in the sense that for a given history, you can find a lot of cells, which is what people in production finds. But at the same time, you have a lower branch that is due to the toxic byproduct of the metabolism. If you remove this connection with the toxicity, that's the kind of phase diagram you find. So you find that the increasing the dilution rate, you increase X. At some point, the dilution rate is so high that you just are removing all the cells, and then you have this big drop here, right? What we found was this this branch that, and then of course you go to the experiment and you say, right, now I understand what is happening. What is happening is that you essentially, are moving up here, and of course if you are here because you were pushing the system to have as many cells as possible, so it's very probable that because of fluctuation you jump from here to here. Right? That was more or less our intuition at the time. And of course that's a toy model. Then we take a realistic model, I would say, from this cellular line, which is, I think, a cancer ovarium cell, something from a hamster, I think. A lot of reaction. So instead of having just 5 reactions, we have like 6,000 reactions. But still, the problem is simple because it's linear programming. And in this S there, instead of having 5 reactions, you have 5,000 and you solve everything. And essentially, if you go only to this plot here, you find the same picture. Increasing X, you have this branch, and with this toxicity, you have this second branch here. Of course, this phase diagram tells you that also for realistic networks, you have something that is consistent with the toy model. More than that, this phase diagram tells you that what's happening here depends a lot on the history of the system. So how you go from one state to the other depends on the story. So if you are here, for example, at this dilution rate at very low X, the dynamics essentially tells you here, and then you can move back the other way around. On the other hand, if you start here, and then this dilution rate with very small X, your system will get stuck here, and the cells will not grow, right? So we published this in 2017, and then we started to make experiments. It's a very hard experiment, at least for a theoretical physicist. You see, this is a cursor that was evolving in time for 90 days. 33.
The previous stuff was also experiment.
No, this is theory. So we repeat the same theory we did before with 5 reactions in a realistic network.
Okay.
Right? So we take, all right, let's try to see if realistic network really works in a similar way, and the picture is consistent. And then we wait to experiment. So we have this collaborator from the Center of Molecular Immunology, so this biotech industry.